Hello and welcome back to the Biochemistry for Health Sciences channel. In today's video, we shall discuss mitochondrial DNA replication. Before we discuss mitochondrial DNA replication, let's remember three very important things about this organelle that we call mitochondria. As we have seen in several videos in the past about metabolism, the mitochondria is where most of the energy in cells that have mitochondria is generated. Recall that most of the energy, or ATP, is generated by oxidative phosphorylation as electrons are transported to the electron transport chain. Another important function of mitochondria is in programmed cell death, especially apoptosis, and we'll talk about this in future videos. And another important function of mitochondria is thermogenesis. The mitochondria generates quite a bit of heat that helps keep our body temperatures at 37 Celsius or 98 Fahrenheit. Another important thing to understand about mitochondria is that mitochondrial morphology is regulated by division as well as fusion. During fusion, smaller mitochondria can join together to form larger ones, while during division, a large mitochondria can split into smaller mitochondria. During fusion, there's mixing of mitochondrial contents, and this allows the healthy mitochondria to merge with those that are partially damaged. Fusion also allows transmission of membrane potential, which is required for ATP generation, throughout the larger interconnected mitochondria. Mitochondrial division helps maintain the number of mitochondria as the cell grows. Division also helps separate the damaged mitochondrial part from the healthy part. So the smaller damaged mitochondria are small enough that they can be basically destroyed by autophagy, or since it's mitochondrial autophagy, we call that mitophagy. Mitochondrial division also promotes apoptosis, because by making more smaller mitochondria, there is a faster release of cytochrome C. And as we'll see in a future video, the release of cytochrome C plays a very important role in apoptosis. Another important aspect of mitochondria is its relationship to several human diseases. This is not surprising, because the mitochondria is the place where most of the energy of a cell is generated, and generating energy 24/7, as we emphasized in previous videos, is extremely important for cell survival. So this mitochondrial division fusion balance, we see this balance being altered in several human diseases, including cancer, metabolic diseases such as diabetes, the process of aging, as well as a variety of neurodegenerative diseases. A little bit about the mitochondrial DNA structure before we talk about replication. Mitochondrial DNA is circular. It has about 17,000 base pairs, which code for 13 messenger RNAs. And these messenger RNAs go on to make protein subunits for complex 1, 3, and 4 of the electron transport chain, as well as protein subunits for ATP synthase in the oxidative phosphorylation. Then the rest of the DNA codes for 22 transfer RNAs, 2 ribosomal RNAs, and then there is also a region here called NCR, or non-coding region. The DNA is double-stranded. One of the strands is called heavy strand or H strand. That's rich in guanine, while the other strand is called the light strand, which is rich in cytosine. Since guanine has a higher molecular weight than cytosine, that's why this guanine-rich strand is called the heavy strand, while the cytosine-rich strand is called the light strand. There are 2 origins of replication, one for the heavy strand, OH, which is in the NCR region, and the other for the light strand, OL, which is around here. Let's look at this non-coding region, or NCR, where the origin for the heavy strand is present, a little bit more. In the non-coding region, or the NCR, there are several features that we need to look at and understand in order to understand replication. We have the LSP, which is the light strand promoter. The LSP is used for transcription of the L strand, and it's also used for primer synthesis in replication. Then we have the HSP here. This is the heavy strand promoter, and this is only used for transcription of the H strand. It's not used for replication. Then we have this CSB, which stands for conserved sequence blocks. There is CSB1, CSB2, as well as CSB3. And we also have this part here called TAS, which stands for Termination Associated Sequence. And this TAS triggers premature termination to form a small DNA called the 7S DNA. So we'll talk about this new H strand, this 7S DNA, at the end of the video. The process of transcription as well as replication are very closely related in the mitochondria. So in transcription, the DNA is copied into RNA, and then the RNA eventually is used to make proteins, and these proteins are necessary for the electron transport chain and the oxidative phosphorylation pathway, which generates energy. The most important source of energy for a cell that has mitochondria is the mitochondria. Now, in replication, this DNA is basically copied into another copy. And then, of course, this copy too can then be transcribed to provide energy for the cell. Okay, so with this background, I think we should be in a position now to talk about the story of human mitochondrial DNA replication. So the story starts from the LSP, the light strand promoter. This is where transcription of the L strand as well as primer synthesis for H strand synthesis starts from the LSP. So the story for replication will start from here as well as transcription of the L strand. But since we are talking about replication here, we will look at how replication is initiated starting from the LSP. Now, this transition between transcription and replication occurs around here, the CSB 1 to 3 region. What exactly controls this? Why does it go through replication, or why does it go through transcription? We don't completely understand. So in an early step, the transcription factor TFAM will bind to the light chain promoter and it will initiate bending of the DNA. The TFAM then will recruit the enzyme mitochondrial RNA polymerase, abbreviated POLRMT, onto the DNA. In the next step, another transcription factor called transcription factor B2 or TFB2M, this will come and bind and unwind the DNA double helix a little bit, and that will give us enough space, and now the POLRMT, the polymerase, will start making RNA either for transcription or as primer for replication. So this POLRMT is used for replication as well as transcription. Mitochondria in humans do not have a dedicated primase that is used for replication. This is used for both transcription as well as replication. So starting from the LSP, this polymerase, POLRMT, will synthesize a new RNA using the light strand as a template. Now, since this light strand is very rich in C, this new RNA here that is made will be very rich in G. As you know, C will pair up with G, so that's why we see this G-rich RNA here. Now, when this new RNA is made past the somewhere around here, the CSB2 region, now the G-rich RNA, this G-rich RNA as well as the G-rich heavy strand, the H strand, they will come together and form this structure here called a G quadruplex structure or G4. And you can see in this structure 4 Gs, 4 guanine residues come together to hydrogen bond with one another to form this G4 quadruplex. This formation of the G quadruplex structure, this G4, will help make what is called the R loop, where the new RNA strand hybridizes with the L strand. You can see that here, displacing the H strand. So that forms what we call a R loop. So this G quadruplex structure together with the R loop will help firmly anchor the RNA transcript. So this new RNA transcript will be nicely attached to the mitochondrial DNA. It has been proposed that during transcription, not replication, during transcription, there is a transcription factor called TFM, and this TFM apparently prevents this G4 formation. So during replication, this protein concentration is very low, so the mitochondria becomes replicated. So in the next step, mitochondrial single-stranded binding proteins, MTSSB, will bind the H strand, the single-stranded H strand, and this will help stabilize the G4 R loop by preventing the re-annealing or rejoining, as well as by preventing attack of the single strands by nucleases. There is also evidence that this MTSSB actually inhibit transcription and instead promote replication by POLRMT. So in the next step, we now have the helicase, mitochondrial helicase, and this is called Twinkle. This helicase is called Twinkle. This will now bind the OH region and start separating the light strand from the heavy strand. So it starts unwinding the DNA. And as the two strands are unwound, once again, the single-stranded binding proteins will come and bind the H strand to prevent it from rejoining to the L strand, as well as prevent it from being attacked by nucleases. It's also believed that these MTSSB proteins also stimulate the helicase, the twinkle's activity. Okay, so now we are ready for the DNA polymerase to start copying the light strand in order to give us the heavy strand. However, at this point in time, the 3' hydroxyl group of the R loop of this primer is not accessible to this DNA polymerase for mitochondria called DNA polymerase gamma. So what happens, there's another enzyme called RNase H1. This will cleave the R loop at multiple sites. Okay, so multiple sites. And that will provide potential primers with accessible 3' hydroxyl groups. So now the polymerase gamma will be able to use one of these sites to start synthesizing the H strand using the R strand as a template. And that's what happens. Polymerase gamma will displace the RNA polymerase, and it will use the light strand, the L strand, as template to extend the primer and continuously synthesize the H strand. Remember, the H strand is copied from the L strand. So as this happens, Twinkle will keep on unwinding the double-stranded DNA. At the same time, the mitochondrial single-stranded binding proteins will bind the free H strand. So initially, only the H strand is copied. So you can see the dashed line here is the new H strand that is being synthesized. So after the H strand synthesis has proceeded for about 11 kilobases, all of this stuff here, the replication machinery now passes the OL region, the origin of replication for the light chain. And as the replication machinery passes the OL, what happens is that it exposes the OL region. The OL in the parent H strand becomes single-stranded, and it folds into a stem loop structure. So you can see the H strand here being folded into a stem loop structure. Now this stem loop structure will block single-stranded binding proteins from binding to this structure. So this will now allow POL-RMT to access a short stretch of the H strand, and it will start initiating RNA primer synthesis. So we see this polymerase now making a short primer using this structure here. So as the POL-RMT binds this loop and makes the RNA primer, this will now initiate L-strand synthesis. So note that H-strand synthesis is a prerequisite for L-strand synthesis. So H-strand synthesis starts first, and only after it reaches this point here will L-strand synthesis begin. So the RNA primer is made for about 25 nucleotides, and then polymerase gamma will come and replace this POL-RMT and will start extending the primer. So the polymerase gamma will now use the H strand as template and it will copy the H strand into a new L strand. And this is done continuously, okay? No Okazaki fragments are made in this case. So unlike what we saw in E. coli or in human DNA replication, there are no Okazaki fragments formed in mitochondrial DNA replication. Anti-helicase twinkle is not necessary for L strand synthesis because the H strand is already single-stranded. So from now on, H strand as well as L strand synthesis will proceed until both strands are almost completed and two daughter molecules are almost formed. So we see the H strand here being copied from the L strand, and then we see the L strand here being copied from the H strand. The primer here at the OH region is removed by nucleases such as RNase H1 as well as exonuclease G, while the primer at the OL region is removed by RNase H1 with the help of another nuclease called MGM-E1. And then finally, once the primers are removed, the two strands are joined together or ligated by the enzyme DNA ligase 3. So at termination, we see a hemicatenane structure that is formed, and this structure have to be separated into two circles. So we have an enzyme called topoisomerase TOP3A. This will separate the two daughter molecules, which have been formed by semi-conservative replication, into two DNA molecules. So this is basically the key steps of how mitochondrial DNA is replicated. Before we end, a few words about something that's very interesting that happens in the replication of mitochondrial DNA. Now, about 95% replication events that start at OH actually don't go all the way to form a circle. They end right here at the termination associated sequence, which is about 650 nucleotides away, to form this short 7S DNA, this new H strand called a 7S DNA. So this 7S DNA is paired with the template with the parental L strand, while this H strand is displaced. This forms what we call a triple-stranded displacement loop that we call the D loop. So you can see the three strands, the parental H strand, the 7S DNA that is pairing with the parental L strand. This is called a D loop that is formed. Why this D loop forms, what's the function of the D loop, we do not really understand. There's a lot of research going on in this area. So that's all for now for this video on mitochondrial DNA replication. Once again, thank you very much for watching, supporting this channel. Have a wonderful day, and we'll see you in the next video.